Good evening. I'm Patricia Van Skype, Director of the Lloyd Library and Museum in Cincinnati, Ohio. I'd like to welcome you to tonight's program, Finding Selective Poison in Plants to Target Breast Cancer. The Lloyd Library brings you this program in conjunction with our exhibition, Pick Your Poison, which was on display the summer of 2024. In a nutshell, the exhibition looks at plants that can do significant harm or, when selective in their use and at the right doses, bring tremendous benefit. Cancer drugs may come to mind. You might be surprised to know that many are derived from compounds found in nature. Dr. Susan Mooberry is well known and respected for her quest to find new treatments for breast cancer, and especially triple-negative breast cancer. Dr. Mooberry is the Professor Emeritus of the University of Texas Health Science Center. She previously held the position of the Greehey Distinguished Chair in Molecular Therapeutics. She's published peer-reviewed articles, book reviews and book chapters, and holds 11 patents on new classes of drug leads. She served as the Principal Investigator for NIH and industry grants and is the past president of the American Society of Pharmacology, where I first met her. The same organization has named her as a fellow, and she was the ASP's first recipient of the Susan Band Horwitz Award. I expect we'll hear more about the award's namesake tonight. Please join me in welcoming esteemed scientist and friend, Susan Mooberry. Hi, Susan.
Hi, Patricia. How are you?
I'm great.
Well, it is such a pleasure to see you, and it's a pleasure to participate in this Lloyd Library exhibition entitled Pick Your Poison. So my goal this evening is to, and in this presentation, is to describe why we look for selective poisons in nature for breast cancer, to describe how we went about it, and then to describe what we found. So it turns out that nature is a prolific source of poisons. And this evening, I am going to define a poison as a chemical produced in nature that has deleterious effects on another organism, just so we're all on the same page. So the ability of plants to inhibit the growth of other plants using chemical defenses has been known for centuries. In the 1st century, Pliny in ancient Rome described the ability of the walnut tree to inhibit the growth of plants beneath the canopy. And we now know that this is due to the ability of walnut trees to produce this compound shown here called juglone. Now, juglone is found in the green parts of the plant. It's found in the leaves and in the stems and in that outer husk of the walnut. And when these fall to the ground, the juglone is released into the soil, and there it inhibits the germination and the growth of other plants around it. So here in the northeast part of the United States, we have the black walnut tree, which is shown there. And once again, like the trees in Europe, it produces juglone. And oftentimes you'll see a walnut tree that is devoid of any plants underneath it. And that's because of this poison. So this provides a survival benefit for the plants because they inhibit the growth of other plants, and therefore they don't have to compete for light and for water and for nutrients in the soil. So numerous other plants, some very common ones, also produce chemicals that have the ability to inhibit the growth of other plants. And some of these include ragweed, the fescue grasses, rice, morning glory, and even sunflowers. And many of these compounds that are produced by plants are very, very complex. Juglone is a very simple example. We also know of other organisms that produce toxins, and this is why we avoid rattlesnakes. Everybody is aware of the poisons produced by the rattlesnake that helps it catch its prey. In the marine environment, there are snails called cone snails, and they produce bioactive peptides. And these peptides actually help these snails in their defense and to capture their prey. And even microorganisms will produce bioactive compounds that are actually poisons against other organisms. So aflatoxins are cancer-inducing compounds that are produced by some Aspergillus species of molds. And many of these are very complex. We think that they're mostly used as chemical defenses, but in some cases we really don't know. But what we do know is that there's a reason why these plants are making these complex molecules. It takes a lot of energy for them to do so. And even if we don't understand why they're making them, there is some reason. So we can say that these are biologically validated. The plant or the snake or the cone snail is making them for some reason that will facilitate the survival of the organism. So we look at the history of pharmaceuticals. Early humans looked around their environments and they used plants to treat symptoms associated with various diseases and with infections. And of course, these indigenous people all around the world had to use trial and error to figure out which things worked and which ones didn't. And over time, they evolved complex pharmacopoeias. And today, some of these are still available as written documents. Much of the world's population still relies to some extent on these medicines. And in many cases, these medicines that were identified by the indigenous people led to our modern pharmaceuticals. So I'd like to tell you some of the stories in the history of pharmaceuticals, starting first with plant-derived drugs. So opium has been used as a pain medication in the treatment of cough and also as an anesthetic for centuries. And the first written record of opium being used was in 3400 BCE. So it is still used today. It is much in the news. We know it's highly effective, but also very, very addictive. Quinine was first identified in the 15th century, and quinine is identified from the bark of the cincoya tree. And it is called the Andean fever tree, and it was used by the indigenous peoples in what is now Peru and Ecuador to treat fevers. When the monks of Spain and Portugal went to the New World, they identified that the bark of the cincoya tree had activity against fevers, including malaria. And so there is actually, historians have indicated that the bark of the cincoya tree that contains quinine was one of the most important exports of the New World because, of course, this impacted the history of Europe by its anti-malarial properties. The source of quinine for over 300 years was the bark of the cincoya tree. And there's a discussion among scholars about the use of quinine and the bark of the tree in the treatment of malaria. And this was an interesting fact that I had never really thought about. Malaria was not a disease of the New World. It was actually a disease that was introduced to the New World by the Portuguese and the Spanish when they imported enslaved people from Africa. So we really don't know historically whether quinine was used first by the indigenous healers or by the monks who recognized this disease, which had prior been known in Africa. But an interesting story nonetheless. Digitalis is a drug still used today that was isolated from the foxglove plant, and that's shown here on the left. The foxglove plant was extracted, and its extract used in the treatment of what was called dropsy, which we now know represents the symptoms of congestive heart failure. But it's really in 1785 when the physician and botanist, Dr. William Withering, published a treatise on the ability of tinctures from the foxglove plant to treat dropsy, and it has been used ever since. Digitalis glycosides are still used today in the treatment of congestive heart failure. There are many newer treatments, but still some people are treated with the cardiac glycosides. Aspirin was available to the public in 1899, but has a long history because aspirin was derived from the compounds that are found in the bark of the willow tree. And a long history is associated with the use of the bark of the willow tree to treat inflammation and to treat fevers. And this was known to people in England. In 1862, Edwin Smith described the ability of this. I'm sorry, it was Edwin Smith in 1862. Oh, I'm sorry, it was, yes. But it was the discovery of some papyrus pharmacopoeia in the early 1800s that really changed the course of the development of aspirin. So what happened at that time is many chemists got involved and they isolated the compounds from aspirin, and then other chemists used chemical modifications to slightly modify them. And aspirin is actually acetyl salicylic acid, where salicylic acid and other related compounds are directly derived from the bark of the Pacific yew tree. Of course, aspirin still is an important drug today, and it is estimated that 40,000 tons of aspirin are produced yearly worldwide. Artemisinin is another anti-malarial that was approved for use in the 1980s, but it's originally isolated from a plant called Artemisia annua. There's a long history of the use of this plant as a ability to inhibit fever, but that information was lost for centuries. And it was really the interest of Dr. Yu Tu who resurrected this information that she had found from an ancient Chinese doctor, Dr. Shi Hong, who had successfully used artemisinin to treat fevers. And so she researched this plant and found the active agent and then actually tested it in clinical trials where it was found to be highly effective. Her efforts to discover artemisinin and get it into the clinic led to her winning the Nobel Prize in Physiology and Medicine in 2015. The vinca alkaloids are extracted from the Madagascar periwinkle plant, shown up there, shown right here. And they were developed in the 1960s as anti-cancer drugs. Two groups of scientists were evaluating these compounds in the late 50s and early 60s. One was a group in Western Ontario, Canada, and the other group was scientists at Eli Lilly. The vinca alkaloids had been used for hundreds of years in the treatment of symptoms associated with diabetes. And when these scientists looked for anti-diabetic effects in extracts from the vinca plant, they did not find any effects. But what they did see was that there was a change in the white blood cells. So this gave them the idea to look at the vinca alkaloids as anti-cancer agents. And I'll talk in a little more detail about them in the next slide, but they're still used today. And paclitaxel is one of the most important drugs that has come into the therapy of cancer in the past 50 years. And it was derived from the bark of the Pacific yew tree. And I'll talk again in a little more detail about that in a few slides. So it turns out plants aren't the only organisms that have led to new drugs. And we have penicillin, which of course was discovered by Alexander Fleming. His bacterial culture got contaminated with a fungus, and he was very observant and noticed that this fungus actually was killing the bacterium. And that led to the isolation of penicillin and really started the antibiotic age. I don't think many of us realize, what life was like before we had all these different antibiotics. And you walk through a cemetery and you see how many people actually died very young due to infectious diseases. So this really was a huge breakthrough. The statins are other drugs that were also isolated from a fungus, and these are very, very important drugs used worldwide to lower cholesterol. Rapamycin is a drug. It's an immune suppressant. It also has anti-cancer activities and is also being investigated in anti-aging implications. It was first isolated in soil from Rapa Nui, hence the name rapamycin. It identified a brand new signaling pathway that we had no idea about until we were able to inhibit with rapamycin. And it's called the TOR pathway, target of rapamycin. And in mammals, it's called the mTOR pathway. And I'll be talking a little bit about that in the next couple slides. And even marine toxins have been identified that have led to new pharmaceuticals. And I was lucky enough to do some of this research years ago, and we were evaluating sponges, and we were thinking that the sponges were the biosynthetic source of many of these compounds. But it turns out it's really the microorganisms that are associated with these sponges and with corals that are the biosynthetic source of many of these marine toxins, which have led to multiple new pharmaceuticals. Now, if we talk specifically about cancer, almost half or slightly less than half of our anti-cancer drugs are still derived from nature. And these are three that I'll talk a little bit about this evening. So the Madagascar periwinkle provides the vinca alkaloids, and I think you can appreciate how complex the structure of these compounds are. And you can see the difference between the major metabolites, vinblastine and vincristine, is only in this one tiny part of the molecule, yet they have different clinical indications and they have different limiting side effects, which is very interesting when you look at the structural complexity and the tiny little difference. So the vinca alkaloids were first evaluated as anti-diabetics. They are then looked at for treatment of multiple different kinds of cancers and are still used today, vinblastine, vincristine, and some semi-synthetic derivatives. These structures, these compounds are so complex that they're not made synthetically today. They are still isolated from the Madagascar periwinkle plant. The story of the development of Taxol, paclitaxel, shows the value of large-scale plant collections that were conducted by the National Cancer Institute. So in the early 1960s, the National Cancer Institute, the NCI, conducted collections, and one of these collections was in the Pacific Northwest, and they collected the bark of the Pacific yew tree, the Taxus brevifolia. And they took that back to the laboratory and extracted it and had some promising activities against the models of cancer that were used at that time. So they sent the bulk material off to Research Triangle Institute in North Carolina, and Drs. Monroe Wall and Mansukh Waney isolated the structure of Taxol from this material. And you can see how incredibly complex this was. And this was really a significant accomplishment. It would have been hard today, even with the tools we have available, to identify the structure, but they did it with much simpler tools. The National Cancer Institute then started testing the purified compound in multiple models of cancer, and it generated some interest, but there was some reluctance because it targeted the same target as the vinca alkaloids. It disrupts cellular microtubules, binds to tubulin. But it was really the work of a scientist at Albert Einstein University, Dr. Susan Band Horwitz. And she evaluated Taxol in her laboratory and found that it actually had a very different, a unique mechanism of action. So both the vinca alkaloids and paclitaxel will disrupt cellular microtubules. Now, microtubules are the railroad system of cells. So these are the structures by which all sorts of nutrients and signal transduction occurs. Now, we know what happens with cancer is that these are cells that have much higher metabolism, and they are oncogene addicted. So everything is happening at a much higher rate. And the microtubules, of course, become very, very important for this heightened signal transduction. So the vinca alkaloids make the microtubules go away, whereas Taxol will actually cause more microtubules to be formed. And they have very different effects on signal transduction, and therefore they have very different activities in patients. So it was really the breakthrough of the identification of a new mechanism of action of Taxol that led to its continued clinical development and ultimately approval, FDA approval for the treatment of ovarian cancer, breast cancer, lung cancer, prostate cancer. There are second and third generation derivatives that are now used. So once again, success story of lots of people involved in it, but some really major breakthroughs. And one more example is a plant that you probably have in the woods around Cincinnati, the mayapple plant. And this plant gave us some lead molecules that were evaluated, but the semi-synthetic derivative, etoposide, is now used as a topoisomerase inhibitor. And it's used in the treatment of multiple different kinds of cancers, including bladder cancer, stomach cancer, as well as lung and prostate cancer. So once again, these are just a few of the drugs that are derived from nature. So I hope I've convinced you that nature has provided many anti-cancer drugs. And I've been lucky over the course of my career to look in many, many different biosynthetic sources for new anti-cancer therapy leads. All of us know cancer impacts us, whether it's in your family or your friends or just acquaintances. We are all very aware that we need new targeted therapies for cancer. We have made huge inroads in the treatment of cancer, and many, many people are cured of their cancer. But still today, for many people, cancer remains a lethal disease. We need to find targeted therapies that will go after the defects that are found in these cancer cells. And we also need to find things that have a much lower toxicity. So that has been the goal during my career before I retired. That was my goal, to find new, effective, and less toxic therapies for the treatment of cancer. And I still believe that there are opportunities to identify new drug leads and/or drug targets in nature, because nature is just a wealth of these very, very complex molecules that are biologically evaluated. So I was able to look in compounds from the marine environment, from cyanobacteria, from higher plants, and then most recently from fungi, to try to pull out these compounds that would have activity that could help in the treatment of cancer. So I'd like to shift gears a little bit here and talk a little bit about breast cancer. So over the course of her lifetime, 1 in 8 women will be diagnosed with breast cancer. And we have made huge, significant advances in the early diagnosis and the treatment of breast cancer. But still today, for many women, breast cancer remains a lethal disease. And we need to identify new therapies to prevent this. There's a pathological classification of breast cancer, and this is how breast cancer is classified. The most common type of breast cancer is ER/PR disease, which is estrogen and progesterone receptor positive. And this represents 60 to 70% of breast cancer patients. 15 to 20% of breast cancer patients are called HER2 positive because they express the HER2 receptor that is found at the cell membrane. And this HER2 is as is the estrogen receptor and progesterone. These are the drivers of these cancers. So the expression of this growth factor is what is causing the cancer. These are the causative signal transduction pathways. And then there's another type of cancer called triple negative breast cancer, abbreviated TNBC. And this name tells you what these cancers are not. They're not ER/PR positive. They're not HER2 positive. They are triple negative, but it doesn't tell you what they are. And that has been the challenge. So we have had a lot of success in targeted therapies for breast cancer, especially with ER/PR positive disease, with the development of endocrine therapy, tamoxifen, and aromatase inhibitors. And what these drugs do is actually prevent the signal transduction that occurs that causes that cancer. So it stops that ER/PR signaling. So it's really getting at the heart of why that cancer occurred. So these are very effective therapies. The similar situation with HER2 positive disease in that the monoclonal antibodies, trastuzumab, target this receptor and prevent its activation. And that's what occurs with trastuzumab and a newer generation pertuzumab. And a very clever idea was to combine the antibody with a cytotoxic warhead. And this is called antibody drug conjugate. And trastuzumab emtansine, TDM1, is a antibody with a cytotoxic warhead attached to it so that the antibody can get to the cells that express HER2 and the cytotoxic warhead can kill them. And it turns out emtansine is a derivative of a natural product. So once again, very, very potent natural product. And there are also kinase inhibitors that have been developed as targeted therapy for HER2 positive disease. For the treatment of triple negative breast cancer, there is only one targeted therapy. And it's not actually targeting the cancer itself, it's targeting the immune system. So pembrolizumab is a drug that will stimulate the immune system to get the patient's immune system to recognize that the cancer is abnormal and that the immune system should take care of it. So once again, a very different type of targeted therapy, not really going after the oncogenic driver. And the challenge to identify targeted therapy for triple negative breast cancers is that it's not one disease. There are multiple diseases shown here by the different colors of triple negative breast cancer. So the one target, one drug approach that has been so successful in the other types of breast cancer will not work for triple negative breast cancer. And that is the challenge of these cancers. In 2011, scientists at Vanderbilt University, led Drs. Lehman and Bauer, identified molecular subtypes of triple negative breast cancer. And this was truly a breakthrough for us in the field of trying to identify new targets to treat triple negative breast cancer. And these are the 5 of the subtypes that they identified: mesenchymal stem-like, basal-like 1, basal-like 2, luminal androgen receptor, and then the mesenchymal. And these are all very, very different, have different properties. They have different oncogenic drivers. And what was the other very, very important thing that Lehman and Bauer did was that they identified cell lines that we have access to in our laboratories that represented the subtypes of triple negative breast cancer. So this allowed us for the first time to screen for distinct compounds that would affect specific to triple negative breast cancer subtypes. So this is really what led to us looking for new drugs for the treatment of triple negative breast cancer. So that led to our drug discovery efforts in triple negative breast cancer subtypes, hence the title, The Selective Poisons, of my talk today. For this project, we used 3 different sources of material. We used plants from our Texas plant collection, and we collaborated with the National Cancer Institute to look at their extract collection. And we also looked at fungi from the Robert Chayovich laboratory. So when I moved to Texas in the summer of 2000, it was very, very hot and very dry, including one day it was 111 degrees. And the ground was desiccated. And I thought, the plants have to be pretty darn tough to survive here. This is a picture of a Blackfoot daisy, which is one of my favorite native plants. And so we began a collection of the plants that thrive in Texas. Many of them are native, but some of them have just adapted to the environment. And we collected and extracted the plants, and you can see these are the extracts right here. So those were part of the plants we looked at in this drug discovery initiative for triple-negative breast cancer. I told you the importance of the NCI collection. We've not discovered Taxol if it hadn't been for that initiative. And the National Cancer Institute has continued their plant collection activities worldwide. So we were lucky enough to partner with them because they use very ethical mechanisms of plant collection. They get permissions. They include the local people in these initiatives. So it's really a model for how these collections should be done. And it's very hard to get the intellectual property arrangements negotiated. So the fact that the National Cancer Institute had done that and then shared with us their extract collection saved us we would not have been able to evaluate these compounds. And we got the NCI collection in these 96 well plates. And then I was lucky enough to work with Robert Chayovich and to screen his fungi collection. He also did all the chemistry involved in this project. And I don't know if you can look at this picture and look up close, and you might look at that and say, those look like Cheerios. Well, you're right. These are Cheerios. And he is so very creative about looking for new sources to grow his fungi on so that they have a very stable matrix so that every time you grow them on the matrix, they produce the same secondary metabolites. So Robert had identified that Cheerios make a really fantastic matrix. We were talking one day, and so Saturday I was working on a grant, and he was about ready to go spelunking into a cave that had an opening about as big as the top of a copy paper box to find new fungi. So once again, very, very creative scientist, and we were able to sample extracts from fungi from very diverse environments, including the soil sediments of the Great Lakes. So these were the sources we looked at for our drug discovery efforts for selective poisons. So hopefully I've convinced you why we looked for selective poisons in nature for breast cancer. And now what I'd like to talk to you about is how did we go about it, and then what did we find? So our approach was to identify extracts that cause selective cell death. And let me describe, what that looks like to us. So we would take an extract here from a plant, and we would evaluate them in 96 well plates where we had plated the cells. But we actually had 5 different plates where we would plate cells of the 5 different types of triple-negative breast cancer. And what we would look for would be the ability of these extracts to kill these breast cancer cells. And this is indicated on this concentration response curve. So here's a concentration here with increasing concentrations off to the right here. And this is percent of control. So as an extract causes more cell death, you will see that the percent of control drops. And what we're really looking for is something that causes this kind of separation. We don't want something that will kill everything. That has been the approach that's been used for the past 70 years. We wanted to do something different and find extracts that would have much more potent effects on 1 or maybe 2 subtypes of triple-negative breast cancer and less potent effects in the other different cell lines. So this is what we were looking for. And it's really this separation that identified extracts that would be targeting an oncogenic driver that was found in some cells, but not in others. So that is what we're looking for in our screening program. The other thing we did is once we identified an active constituent, we used the chemistry and our biological assays to do bioassay guided fractionation. And this required very close coordination between my laboratory and the Chayevich laboratory, and it worked very, very well. Many times in natural products, chemistry will be the driver, and what chemists are looking for is a very interesting new molecule. That was not our goal. If that happened, that would be fine, but we were focused on trying to find new therapies for cancer. That was our goal. It wasn't to find new chemistry. So everybody on the team was very committed to that. And as I say, it worked very, very well. So we would find an extract that had this kind of activity, this separation. And then we would send the extract to our chemistry collaborators who would fraction them. And oftentimes we would get 9 to 11 fractions per extract. And most often we would see this kind of profile where we didn't have any activity at all. And we knew that there was nothing there that we were interested in. And hopefully we would find a fraction where we had recapitulated what we saw with the crude extract. And that's what we were looking for. It's shown here with fraction 2. Sometimes we would get a fraction where we had a little bit of activity, but once again, there is no separation here among the different breast cancer cell lines. So that wasn't what we were interested in. And we were able to do this and pretty much in real time, because you can't also leave your chemists hanging without giving them results. So they would send the fractions to us late in the week. We would put them on the cells and the next week on Tuesday morning. We would get the results on Thursday, and we would make sure that they were in their hands by Friday. So nobody was waiting around for bioassay results. And it was a commitment on the entire team, and it really did work very well. So what did we find? Well, I'm going to tell you 3 brief stories of 3 of the things that we found as a result of this project. I'll tell you about digualin that we found from Amorpha fruticosa, the false indigo plant, which is a Texas native. From the NCI collection, we evaluated Desmanthonium guatemalase, and others, such as sunflower, which provided the fercarinol compounds, and the most potent one, which was dihydrofercarinol. And then a story about keeping up with your colleagues. And as I said, this was a collaboration with the National Cancer Institute. And I was visiting the National Cancer Institute in Frederick, Maryland, and John Butler talked to me and he said, you know, we have this very interesting compound we got out of a Tanzanian plant, and it has some really specific activities against certain kinds of cancer. Do you want to look at it? And I said, sure. So that's the story of unglerin A and its effects in some triple-negative breast cancer cell lines. So here was one of our first selective extracts. This was from our Texas plant collection, and this was a picture taken by Paul Cox, who was the superintendent of the San Antonio Botanical Garden, and that's where we got most of our plant material from, and he was a really fantastic partner. And what we saw here with this extract, number 205L1, was a separation of the curves. And this was work that was conducted by a graduate student at the time, who is now an assistant professor, Dr. Andrew Robles. And this is the kind of activity we were looking for, a separation here between these different cell lines. And the most sensitive cell line here is the MDA-MB453, and that represents the luminal androgen receptor subtype of triple-negative breast cancer. So with our chemistry colleagues, Robert Chayevich and Sheng Kai, they isolated the natural product digualin, which was not a new compound, had previously been described as the active agent. And you can see that the digualin totally recapitulated what we saw in the crude extract, and it inhibits the growth of these luminal androgen receptor cells with a 33-fold selectivity for this cell line as compared to the other types of triple negative breast cancer. So then Andrew embarked on trying to figure out what the mechanism of action of degwalin was, and he just was very creative. He looked to the driver pathways that we know are effective in causing the luminal androgen receptor to be the oncogenic driver pathways. And one of them is the PI3 kinase AKT-mTOR pathway. So this is that mammalian target of rapamycin pathway. And so he looked at various levels of this pathway and found that degwalin inhibited the ability of mTORC1 to phosphorylate its downstream substrate. So you can see where these X's are is where we saw activity of degwalin inhibiting this pathway. And this happened within an hour with very low concentrations of degwalin. So we were convinced that this was probably part of its mechanism of action. But the mTOR inhibitor rapamycin did not have the same profile in that cell line, so we knew that there was something else going on too. So then Andrew looked at the androgen receptor. I mean, these cells are driven in part by the high levels of expression of the androgen receptor. And he looked at the levels of the androgen receptor following treatment with degwalin and saw that he got inhibition, about a 30% inhibition in the levels of the androgen receptor. He then also looked at androgen signaling and found that the ability of androgen to move into the nucleus and to stimulate cell proliferation was also inhibited to a similar extent. So it looks like what's happening with degwalin, even though it's not a drug lead, it doesn't really have great pharmacological properties that would cause it to be an optimal drug. It informed of a combination of an mTOR inhibitor and an androgen receptor inhibition in the LAR subtype of triple negative breast cancer. Andrew looked at this combination in vitro in the cell lines, and it was additive, taking 2 FDA-approved drugs that target these pathways, and he did see additive activity. But when he went into a murine model of cancer, he did not see that activity. And it turns out clinicians had also tried, were at the same time trying this in the clinic, combining an mTOR inhibitor, rapamycin, with an androgen blockade drug that is used in the treatment of prostate cancer. So they evaluated this in the LAR subtype of triple negative breast cancer, but it really was not effective. So really good idea here, but it just didn't pan out that we got a drug lead or really a new way to treat this luminal androgen receptor subtype of triple negative breast cancer. We evaluated an extract of Desmethonium guatemalase from the NCI, and this was a plant that was collected by the NCI in Guatemala. And you can see here that we had a separation of the curves, and one of the cell line that was the most sensitive was the MDA-MB231 cell line. We were very excited about this because this is really a very, very aggressive cell line, probably the most aggressive cell line of our panel. So we were excited to see this kind of profile. So then we continued evaluating this, and we identified 6 fercarinol compounds from that Guatemalan plant. And the most potent one is dihydrofercarinol. We also isolated other fercarinols from related species. And this was work that was conducted by Karina Grant in my laboratory and Dr. Sheng-Xing Kai in Dr. Cheovich's laboratory. And Sheng-Xing was very creative. He looked into the literature and saw that the falcarinols were isolated from sunflowers. So he started growing sunflowers at the University of Oklahoma, and then he harvested them and isolated another 6 or 7 falcarinol compounds. And you can see here, we were able to get structure-activity relationships from having so many compounds. But you can see that the two cell lines now that represent the this mesenchymal stem-like, the MDA MB231 and the MDA MB157, were the most sensitive of our cell line panel, with 5 to 22-fold selectivity for these cells. So now the question was for Karina, how does this work? We knew from this compound that we didn't have really great pharmacological properties, so we knew we had to identify the mechanism of action. So she did that. She was very strategic. She first investigated the Lehman and Bauer predicted driver pathways and had no possibility that that was how this was working. She looked into biochemical and affinity methods, but once again, that really was not going to work for this compound because of the structural features of it. So that led her to a genetic approach. And what she did with the help of our colleague in the laboratory, Dr. April Reisinger, used a CRISPR-Cas9 knockout screen. So essentially they knocked out every single gene in the cells and then found out how knocking down one gene would lead the cells to survive. So it was a genetic screen. We had help from the Broad Institute and John Dorsch. And it turns out we were very lucky in that when we look at this volcano plot, there was one gene, and the screen knocked out that gene. These cells became insensitive to the dihydrofuranol. So this one gene, and it was 17 beta hydroxysteroid dehydrogenase type 11, a gene product, a protein that I had never heard of before. So we had to investigate, what this enzyme did. So we first looked to see, well, is it even expressed in our sensitive cell line? And yes, it is. We have the highest level of expression in the cell line that is most sensitive. We used siRNA to knock down the levels of this protein, and that actually reduced the sensitivity of the cells to this protein. So that, once again, was very, very consistent with its activity. We then tried to introduce the gene into cells, into this BT549 cell line, to see if that gene itself would allow those cells to become very sensitive to the gene. And in that case, it didn't work. So that gave us an idea that in part, the MDA MB231 and the 157 cells are sensitive due to the expression of this protein, but that's not everything. So it's a much more complicated story than just one gene and leading to sensitivity. So it was not a promising drug lead chemically, but there are some remaining questions. How does this enzyme confer sensitivity to dihydrofuranol? What other proteins are important for its activity? And we haven't answered this yet, but it is really remains a very interesting question. And for the last story, I'll tell you about unglerin A. So this was a compound that was first isolated in 2009 by the National Cancer Institute from an African plant, Phyllanthus ungleri. And they found that it had highly selective actions against some cell types, including renal cancer cells. So as I said, Dr. Butler mailed it to us, and we looked at it in our triple negative breast cancer panel. And here we found what we had hoped to find throughout our screening. And we found one cell line that was just exquisitely sensitive to this compound, whereas the others, were much less sensitive. So you can see the separation here. Their BT549 cells are 1,700-fold more sensitive. So, that was very, very promising. So work conducted by the National Cancer Institute and their colleagues had identified that unglerin A is a TRPC channel agonist. So these TRPC channels are channels that are found in the membrane of cells, And an agonist is something that will turn them on. So if one puts unglerin A in the media outside the cells here, this will open up these channels and allow ions, including most important calcium, to flow into these through these channels into the interior of the cells. So that was its mechanism of action in renal cells. So we asked the question whether this was also happening in our sensitive cell line. And it turns out that you first have to express the protein, and yes, the BT549 cells have a very high expression of these TRP1/4 channels. So then Karina looked at whether unglerin A would cause a calcium influx, because if that's the mechanism of action, you should be able to see that. So she loaded her cells with a calcium sensitive dye and evaluated them microscopically. She treated with a low concentration of unglerin A and then imaged every 30 seconds for 3 minutes. And this was the result. After 30 seconds, you can see the baseline, those little green dots are the cells here. And then within 30 seconds, with 100 nanomolar concentration of unglerin A, these cells were just glowing in the media. So what was happening, this is indicative of calcium rushing in from the outside into the inside of the cell. And what happens is that this causes membrane depolarization and rapid cell death. And that's exactly what was happening in these BT549 cells. So unglerin A for us to find a new target for a subset of triple negative breast cancer cells that are defective in handling changes in intracellular calcium. And these are cells that express high levels of these TRP1/4 channels. And this actually opened up the possibility of using other cation disruptors in treating some types of triple negative breast cancer. So if you think back to the digitalis glycosides, these actually do inhibit cations, and they inhibit sodium potassium ATPase. So this is their mechanism of action. And it turns out that this cell line, the BT549, are exquisitely sensitive to the digitalis glycosides. So then Karina and another graduate student in the laboratory started digging into the literature. And there is some literature showing that some women who are undergoing treatment for breast cancer and also have congestive heart failure and are being treated with the digitalis glycosides, those that have triple negative breast cancer had a longer time to disease progression. So there is just a little hint in the literature that potentially for some women that have high levels of expression of these TRP1/4 channels, that potentially a cation disruptor might have some utility. So some clinicians have looked into that, I believe at MD Anderson Cancer Center. So I hope I've convinced you that nature continues to provide us with some selective poisons that could be used for new leads for the treatment of cancer. And some of these will be new drug leads, and others will identify pathways that can be targeted with FDA-approved pharmaceuticals. Remember, our goal is to make an impact for patients, so if we can get there faster with FDA-approved pharmaceuticals, that is really the best way to go. Well, none of this would have been possible without a huge team of people that I was lucky enough to work with. So my two major collaborators that I really want to shout out to, Dr. April Reisinger, who worked together with me on this project over the course of 15 years. And she is now an associate professor, tenured at the University of Texas Health Science Center at San Antonio. And then my collaborator on this project, who provided us the fungal samples and who did all the chemistry, Dr. Robert Chayovich. He was at the University of Oklahoma. I called him last week just to check to make sure I could show his pictures of fungi, which he readily agreed to. And he informed me that he was just moving into his house in Ann Arbor, and he has accepted a position at the University of Michigan and will be continuing his work there. And then in my laboratory, Andrew Robles, Karina Grant are fantastic screening technician, Cora Peterson, who made sure that the data got to our chemistry collaborators in a week, Dr. Sheng Kai, who did much of the chemistry, our NCI collaborator, John Butler, and Barry O'Keefe and Tanya Grokovic, who were also at the NCI, who provided us with the plant extracts. John Dosh helped us. He's at the Broad Institute with our CRISPR-Cas9 screen. Paul Cox was fantastic help in our Texas plant collection. He was a superintendent of the San Antonio Botanical Garden. Evelyn Jackson, Lauren Clark, and Mildred Obadopke did the early early plant collections. Rachel Hartley Foster was just a fantastic she jumped in learning the chemistry of some of our early collections and really started us off on our project on the Texas plants. Andrew Robles was involved also in the plant collections, and Jianan Peng was our chemist who did the early chemistry. And then there are many, many others that is just too numerous to list. We had support for this project from the National Cancer Institute with a cooperative U01 agreement and my Greehey Endowment. And now I would like to thank you for your attention for this presentation.
Thank you, Susan. Oh, I've got my video off again. Thank you, Susan. That was really fascinating. Get a lot of comments, great work and presentation. Very early on, Jen said that she really appreciated going through all of the plant history. I do want to point out that at the Pick Your Poison exhibition, we have digitalis, and we've also looked at things like quinine too. So it's a really nice sort of overlap. Michelle asks, are there plants that combat estrogen-based breast cancer?
So the early, I have to tell you, this is not my area of expertise. You know, I have really focused only on triple negative breast cancer. I am not knowledgeable about the estrogens. You know, there are many estrogens in our foods. So for example, tofu has estrogens. And there's a huge debate whether this is a beneficial effect, if there are so much in there that it could be a driver for breast cancer. There's a lot of literature out there. I really don't know the literature, and I can't give an informed opinion on that. But there's a lot of literature out there about, you know, multiple plants producing estrogenic compounds, and there's a big discussion about whether they could be beneficial or whether they could have a negative effect.
And then I have a question that's you know, step back, you know, 100 from the clouds sort of look. You know, you mentioned that 50% of cancer drugs were nature derived. And that was something when I first started at the Lloyd Library and before I started hanging around with Pharmacognosy, that, you know, that was a complete shock and very new to me. And I think, you know, even like tonight, you talked about the statins. I had no idea that statins were derived from microorganisms. Why do you think it is that we have a blind spot, or that we're not aware that nature plays such an important role in these drug discoveries?
I think we all tend to think that the newest technology is the best. So things that are new have to be better than what's old. So even in some of the articles I read, you know, there was misinformation that the fact that these were synthesized and not that they're derived still from nature. So, you know, I think it's the fact that we all jump on the bandwagon is that new has to be better, and that's not the case. And we need to look back at what has been successful and continue that, as well as advance the new ideas.
So, and I know this is probably a very, very broad stroke and simple question, but you mentioned some plants from different parts of the world, Guatemala, Africa. And And I understand that the compounds just target a certain part of the cancer and all of that. But do you see any decrease or less incidence of breast cancer in areas where these plants are rampant and native?
We don't.
Okay.
And breast cancer is very, very complex. And I was lucky enough to be at the University of Hawaii with a very big epidemiology group. And, they found that there was a low rate of cancer in Japan, but as the Japanese Americans moved to Hawaii and adapted a diet that was very similar to others, that their breast cancer rates increased. So it's so much more than just the environment. It is, what we eat, and there are other pressures. And if people are dying of infectious diseases, then they're not going to reach an age where we normally would diagnose breast cancer. So it's very, very, complex.
And I know with Taxol, for example, it's just a microscopic part of the compound. So obviously, if you happen to walk by a Pacific U, you're not going to have your risk of breast cancer go down.
Yeah. And as you mentioned earlier, it all matters is the dose, right? And, what some of the advances that were derived, some of the advances that led to these pharmaceuticals was really pulling out what the active agents were and getting rid of those that caused the side effects, and really defining what the appropriate dose was. So those were very important things that led to these development of pharmaceuticals from the information from indigenous peoples.
We have a question from Jen saying she'd love to know if you're familiar with Chantrell Cabrera, a registered herbalist with the American Herbalist Guild, doing tons of work with patients alongside doctors using plants.
There are some people doing really fantastic work along that line. It's not what we did. We didn't want to compete in that space because we knew there were people already doing that. So what we did was try to avoid plants that we knew other people had already looked at. So, we weren't trying to, they're the experts in that area. And we tried to do something very, very different and avoid plants that we knew other people had looked at. And yes, there are some people doing excellent work along those lines. I don't know them specifically. I've never really kept track of that because we had a different approach. It was complementary, but not overlapping.
Well, Susan, thank you so much. I'd like to also thank our audience for joining us. Watch for tonight's program to be posted on the Lloyd's YouTube channel, and we hope you'll share it with people as well. I'd like to point out that programs like these are supported in part by members of the Lloyd Library. If you're a member, thank you very much. If you're not a member, I encourage you to join, and membership information can be found on our website. We also encourage you to visit our social media, and especially our Instagram feed, which is Lloyd Library Official. Again, thank you all and have a great night.